Welcome back. Proteins, part 2. All right. So proteins can be divided into domains, and a domain is like a chunk of a protein. Think of a protein as like a train. Can you picture a train? You get all those cars, and each car is like a place where you can sit and things happen. And then there's like a little connector in between. And you could put more cars on or fewer cars on, and it would still be a train as long as it's got an engine. Then it's a train. So it's kind of the same with proteins. They tend to be organized in domains. The domains are like the cars of a train. They're like independent little units. And so when these things evolve, there really aren't any good mechanisms inside of living things that will invent a domain. That's actually very hard to do. Or rather, it would take you millions, maybe billions of years of trying to invent a working domain of some kind. But stealing domains is definitely a thing that happens all the time. So while there are an infinite number of ways that you could assemble proteins, there's a relatively small number of ways you can assemble proteins that life has found and actually found a use for. And so these are the useful protein domains. And so if you know what protein domain you're looking at, there are probably only a few kinds of protein that actually use that domain, have that shape. And so that's a big advantage if you can break down the domain structure of your protein. So now, how big is a domain? Well, in a prokaryote, so like bacteria and archaea, average size is around 100 amino acids. And so if you think about that, how big would that be? Well, 100 amino acids needs 300 nucleotides. So proteins are usually not going to be smaller than one domain. As you can see here, there are very few that are smaller than, around 100. That's where it really starts getting interesting. And there are things that are bigger. So if we're looking for genes, we're probably going to look for something that's 300 nucleotides and an open reading frame. That's kind of on the small end of what a gene is, and it can go bigger. Now, if we're talking about eukaryotes, they tend to have larger genes, or rather the domains of their proteins are going to tend to be about 150, something like that. When proteins get larger, they usually don't just increase in size everywhere. Usually they get larger by taking one domain and attaching another domain to it. And so let's say this domain is an enzyme and this domain sticks to something. If you join them together, it's just like Legos. Now you have something that sticks and is an enzyme, and so it'll drive enzyme activity to a particular part of the cell. That's how evolution works. So we just take these parts that already exist, combine them in slightly different ways, and if it works and you don't die, then boom, you pass it on to your kids. Yeah, fantastic. All right. So if that's domains, let's talk about entire genes, because genes can make a multi-domain protein. And here we're pretending that genes only means things that encode protein, when we know, between you and me, that there are genes for tRNAs and ribosomal RNAs and all kinds of other stuff. Pardon. But for the most part, genes and proteins, let's assume those two go together. So, average size of a gene in prokaryotes, about 300 nucleotides. So 300 nucleotides, how many amino acids does that make? About 100. Yeah, you're right. So about how many domains do you find in a standard prokaryotic gene? One, right? Yeah. Yeah, because each domain in bacteria and archaea is about 100 amino acids. That's like the average size. Okay, so for eukaryotes, what's the average size of a gene? Around 450 to 600. How many domains do you have in the average eukaryotic gene? 1 to 2. Yeah, there you go. And you'll see there's a long tail out here. These are the really big genes, and there are genes out there that'll have 20 domains in them. They're really fantastic things, very complicated, like an entire assembly line all in a row, working together toward some job. Yeah. All right. So let's learn a little more about these domains. So, all right, what's a domain? So it's a smaller part of a protein. Okay. But there are different kinds of domains. And so up to now, I've just said the word domain as if, that just means one thing. That is not the case. I'm sorry for misleading you like that. That's not very nice of me. So there are different ways to define a domain. You can define it as a functional domain, which means kind of the minimum amount of protein that'll actually do a job. If it's an enzyme, this is like the smallest version of that enzyme that still does the enzyme thing. Yeah. If it interacts with something, it's the smallest part that'll still interact with that thing. Quite often, even though only a small part of a protein may be involved in an activity, all the rest of the protein has to be here to hold everything in just the right geometry so that those little parts of the protein are exactly in the right spot in order to be able to do their job. Proteins are very, very precise. That's why they work so well. So they have a very precise structure. And they'll have some parts that are very rigid. So like these big lumps here. This is a pyruvate kinase. The green lump and the white lump and the blue lump, those are all probably very rigid. They probably do not flex much. But in between them, you see this sort of floppy blue loop? That's probably bendy. So these are like the 3 train cars, and the sequence in between is like the little connector between the train cars. It's a flexible linker. That's what we call it. That's how proteins work. Multi-domain proteins anyway. All right. So there's functional domains and then there's structural domains. To actually know what the structural domains are, you have to solve the structure, unfortunately. And so that means solving the crystal structure or NMR structure or cryo-EM structure, something like that. And generally, that is going to take years and many thousands or tens of thousands of dollars. And so usually that's going to be not something that you do. The people that do this are very specialized. That is all they do for their entire careers. And they learn really cool things. But yeah, that is the one thing. It's a real specialty. So yeah, structural domains, neat if you happen to have the structure. But if you don't have the structure, that's kind of off the table. And functional domain, you have to actually confirm that experimentally to know for sure if it is. And so if you're dealing with something that's brand new, you do not have that to fall back on. And so if we're talking about new things, which is where bioinformatics is really, I think, interesting and useful, then the best we're going to get are phylogenetic domains, or domains based on conservation. Domains based on parts of the protein that don't change very much compared to the parts around them. And so how many phylogenetic domains do we have here? Well, let's go see. Actually, no, I left out that slide. So it turns out this is, I believe, 2 phylogenetic domains. I believe this part here tends to always occur together, and I think this part is sometimes present, if I'm reading that correctly. But yeah, that's the idea. Okay. So, what are our criteria for deciding if something is conserved? Because saying something is conserved is saying that it shares a common ancestor with something else. And not an ancestor as in terms of like the whole organism, but an ancestor in terms of that gene or the protein that gene makes. If we can show that 2 things are related beyond a statistical, reasonable doubt, then we can do a lot of stuff. We can build family trees out of them. We can make a multiple sequence alignment. We can analyze them. And if we know the function of any one of the things that's related to this, then we can assume quite reasonably that the function should be conserved. And most likely it will be. So that's really nice. So here's the order of evidence. So what changes fastest versus what changes slowest? So the fastest changing thing is nucleic acid. So like the DNA or the RNA that encodes a protein. Why do I say this changes fast? Well, if you've ever looked at the coding wheel, and I mean really looked at it, you'll notice there are only 20 amino acids and 3 stops. So 21 things, if you like, and there are 64 codons, which means that for most amino acids, there are multiple ways that you can spell. You can say, put in this amino acid with a particular, combination of DNA. So you can change certain parts of the genetic code, and it won't change the machine that you're making. And so, yeah, the genes wander quickly, but yeah, the other things change more slowly because if the gene changes in a way that actually breaks the protein, and if the protein was important for keeping the organism alive, then a genetic change leads to a dead organism, and so those get filtered out real fast by natural selection. All right, so gene change is fastest, but if we can find a genetic match that's statistically significant, then that is the best evidence of all. We can guarantee that these two are, to the maximum extent possible, they're going to be the same in every other way. So they're going to have the same primary structure, like structure of, rather the amino acid sequence, same secondary structure, or at least related secondary structure, like alpha helix, beta strand, that sort of thing. They should have a lot of the same features, like same number of transmembranes in the same position, same amount of glycosylation, signal peptides, amphipathic helices, that sort of thing. They'll probably have the same function, and they will definitely have the same fold, which is like the overall three-dimensional structure of the protein that they make. So if you can get one of these high criteria, if you can find a match there, everything below, you can assume that is true. So that's what it says here. At stages 6, 5, and 4, if you can find a statistically significant match, then you can infer that 3, 2, and 1 are also going to be the same. And it's not guaranteed because nothing in stats and nothing in bioinformatics is guaranteed, but that is the best, most solid inference you can make. So we'd call that definitive evidence. And so, all right, so how do you get this definitive evidence? Well, you can do something like a BLAST-N. That will look for nucleic acid matches. So that's a nucleotide blast, is the other way to say that. If you want to see, does the protein match? Because the nucleotides change, but the protein is going to be more conservative. It's more slow moving. You can do any one of these things, like BLAST-P, BLAST-X, or T-BLAST-N. Those are all ways of comparing protein to protein or protein to nucleic acid as a way of just sort of slowing down evolution and taking a broader view. Or HH-Blitz will actually do that very nicely. It looks at amino acid sequence and a little bit at amino acid structure. Or if you want to really ramp it up, so if you're not finding anything on BLAST-N and you're not finding anything on this level, then the next step is you go to HH-Pred. And so it's going to look at both the structure and the sequence at the same time, and it's going to try to find a match. So this is very good at finding distant matches and things that have had a lot of time to evolve and have changed quite a lot. If you can find a hit there, that's great. Any one of these is good enough to infer all the other things below. Now, let's say you can't get a hit on 6, 5, or 4. Unfortunate. But there's always supporting evidence. So if you can show that you have a lot of the same features, it's not definitive, but it's suggestive. Yeah. And so this would be like saying that the context is the same. We've got this thing at the beginning, this thing in the middle, this thing at the end. Can't find anything else in common, but they actually could be related. So it's a way of making a hypothesis that's not completely useless. Let's put it that way. If you can show they have the same function, that's wonderful, but function is very hard to predict. All we can really predict are sequence and maybe structure with sequence at the same time. There isn't a good way to predict function beyond saying that it has the same structure as a thing that has a particular function. Yeah. But if we happen to know for some reason that this gene has a particular function, like you've got it out and tested it, awesome. Yeah. So if it has the same function, you can infer that it probably has the same fold, although not every time, but most commonly. And then fold, you will find that fold is the last thing to go. So if you have two things that are related and they diverge over time, first the genes won't look similar, then the amino acids won't look similar, then the secondary structure will start to look different. Then after that, some of the features will start to go away. And after that, I mean, function is probably preserved up to that point, but then function's going to kind of probably change or fall apart. but you still may be able to find that they are similar folds. And if they are, then that's good grounds for saying that they are evolutionarily related. Problem is, you can't predict fold very easily, or rather our fold prediction methods are worse than our prediction methods for everything else. And so they're less reliable, and so it's not something you're ever going to be able to say is definitive. And you're probably not going to have the three-dimensional structure of a new thing if you're trying bioinformatics on a new thing. So it's just not on the table, but if it was, boy, that would be the best evidence. So yeah, it's desirable, but probably unattainable. All right. So there you go. That's a little primer on how to think about protein, how to handle proteins when doing bioinformatics. So thanks very much.